chill beats with lights, smooth piano melodies, soft synth pads, mid-tempo groove, clean and minimal, modern African tech vibe, relaxing but intelligent mood, perfect, no vocals. Afrofuturistic chill beat with light, percussion, smooth piano, melodies, soft synth pads, mid-tempo groove, clean and minimal, modern African tech vibe, relaxing but intelligent mood, perfect background for educational content, no vocals. Afrofuturistic. Afrofuturistic chill beat. Cooking up in your mother African divide. The rest of my things like a jello. If it don't come for the tuition, one then no for life, matter and control my education. And I got to make that. I thought you is fine. Innovation. And I just thought, hey, 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 hey. I'm the truth. Oh, I thought you. I thought you. I thought you this time. Innovation, innovation. I thought you. I thought you. I thought you this time. Innovation, innovation. I thought you. I thought you. Afrofuturistic shimmy like the thousand. So the many sense some plastic tattoo. When you make me move, I can laugh at the fire. Relaxing what the time become on my papa. If you just laugh, take it so tight, take that focus. Or don't then focus, no TV vision content. Indonesia, I felt you respected. Indonesia. Nothing was like getting more Asia. Nothing was like getting more Asia. Nothing was like getting more Asia. Epic cinematic, futuristic science theme, emotional and inspiring. Deep bass pulses with rising ultra-terrestrial strings. Modern tech atmosphere, similar to Hans Zimmer, meets documentary science soundtrack. Slow, build intro, then powerful drop. Evops discovery. DNA, space, and innovation. No focus, high innovation. Perfect for educational video. Epic cinematic. Epic Cinepigeons Mensen. Futuristic science stimmen. Emotional and inspiring. Deep dives pulsate with rising Augustus James. Cetoetonics and flyers. Both entire atmosphere. Similar to hands emanates ducking and desired soundtrack. Slow go intro then powerful jaw. Evokes the stuffily DNA, space and innovation. No vocals, energy climax. Clicks for fiver. Intro timex.
For decades, analyzing the building blocks of life required
Slow go intro then powerful jaw. Evokes the stuffily DNA, space and innovation. No vocals, energy climax. Clicks for fiver. Intro timex.
So, good evening once again. I want you to continue. This is just a 5 to 6 minute video. It's also going to tell us what we want to do today so that you continue viewing and start taking notes. Thank you.
For decades, analyzing the building blocks of life required and the initial DNA sequence produces a full. For decades, analyzing the building blocks of life required access to million-dollar laboratories filled with specialized equipment. Today, that work can be done from almost anywhere. By the end of this session, you will manually translate a raw genetic sequence, identify an unknown organism using a global database, and document your findings in a professional lab report. This process of gene expression, how a cell reads genetic instructions to build life, is the data pipeline that powers modern biological research and computational biology. To execute this pipeline yourself, you only need 4 things: a smartphone or
So, good evening once again. I want you to continue. This is just a 5 to 6 minute video. It's also going to tell us what we want to do today so that you continue
initial DNA sequence produces a of biology to a practitioner of computational science. Required access to million-dollar laboratories filled with special and uses the chemical bases adenine, thymine, guanine, and cytosine. On the right, RNA is single-stranded, and it swaps out thymine for a base called uracil. The biological pipeline flows in one direction. The cell reads DNA instructions to build an RNA messenger. That RNA then translates into a chain of amino acids, which finally folds into a complex three-dimensional protein to perform specific jobs in the body. When this pipeline misfires, the physical consequences are immediate. Sickle cell disease is a clear example of this sequence breaking down. In sickle cell disease, a single incorrect base letter in the initial DNA sequence produces a faulty RNA strand. That faulty RNA translates into the wrong amino acid, causing the final hemoglobin protein to fold incorrectly and warp the shape of red blood cells. A single character swap in a microscopic sequence directly dictates biological function and fundamentally shapes a patient's health. Step 1 is to write down our raw starting material. Open your notebook and write this exact DNA sequence in block letters: ATT, CGA, TTT, GAA. Now we need to transcribe this into RNA. Remember the rule for RNA. It uses uracil instead of thymine. Every T in your DNA sequence must become a U. Go ahead and write the matching RNA sequence directly below your DNA letters. You should now have AUG, CGA, UUU, and GAA. This chart is a codon table, which acts as a translation dictionary for cells. By looking up our RNA triplets, or codons, we can find the matching amino acids. AUG codes for methionine, CGA brings in arginine, UUU translates to phenylalanine, and GAA provides glutamate. By converting those raw letters into a specific chain of amino acids, you have manually performed gene expression. You decoded the instruction set for a protein by hand. Now we need to figure out where this sequence came from. Open your mobile browser and navigate to the NCBI BLAST website. NCBI stands for the National Center for Biotechnology Information. Click on the Nucleotide BLAST tool to compare our sequence against their records. Paste your original DNA sequence, ATG, CGA, TTT, GAA, into the large query box at the top of the page. Scroll to the bottom and tap the BLAST button to execute the search. While the page loads, an algorithm is performing sequence alignment in the background. It compares your endpoint against millions of stored sequences, adding points for exact letter matches and subtracting points for mismatches or empty gaps. When your results appear, look for the E-value. The E-value, or expect value, is a metric that measures the statistical significance of your match. This scale shows how to read your results. A lower number, like 0.001, indicates a much stronger match because it means there is almost zero probability that the match happened by random chance. A high number, like 10, points to a weak, likely random match. By filtering the results through the E-value, scientists can cut through millions of irrelevant database entries and confidently identify genetic material. Look at the top row of your mobile results screen. Identify the scientific name of your top match organism and record its specific E-value. This diagram illustrates a phylogenetic tree, mapping how organisms relate to one another over time. By analyzing the sequence similarities and differences found in databases like BLAST, researchers can chart shared ancestry and track how specific mutations cause species to diverge. With our organism identified, it is time to formalize our findings. Open the Bioinformatics practical report template provided for this session. Fill in the aim of our experiment, list the materials you used, outline your procedure, and record your results, including the organism name and E-value. Finally, complete the interpretation section by explaining your match strength, and use the reflection section to write down what you learned and what challenged you during the process. Formalizing your observations is a baseline requirement for scientific credibility. Following this template ensures your results are organized for peer review and further research. In one session, you progressed from translating raw DNA letters on paper to tracing evolutionary origins using a global database. Your completed Bioinformatics practical report is the result of that work, a record of your ability to navigate the digital tools of modern genomics. You can continue this exploration. Try inputting a random string of DNA letters into BLAST to see if you can find segments of human, viral, or plant genomes. Share your lowest E values and the organisms you discovered in the comments below, and let's see who can find the most statistically significant match. Accessing these global data repositories from any device means you can practice modern genomics outside of a traditional lab. You are now equipped to navigate the same databases used by researchers worldwide.
So that is the summary of what we want to do today. I said that we are going to do what is called blast. Blast. Blast means basic local alignment sequence tools. We are going to see it. Now let's begin um, this practical journey. Don't forget that all what we have been saying so far is to visualize our DNA in form of letters, which is a code, 4-letter words. And from that video, you will see that the 4-letter codes are adenine, thymine, guanine, and cytosine. That is in DNA. And in RNA, the difference between the codes, the nucleotide code in DNA and RNA is that this T has been replaced by another nucleotide base which is called uracil. Now there is something we call the Central Dogma theory. Central Dogma theory is the genetic flow of information from double-stranded DNA to single-stranded RNA, then from RNA to protein, that is to amino acid. That is the flow. And just like I told you in the last, in the first lecture on bioinformatics, I told you that you need to start learning about the informational molecule. And bioinformatics is nothing but how to decode, how to explain, how to analyze, and how to retrieve the whole information about organism, the whole organism in form of codes, in form of letters. And that's why today's topic is titled the Digital Lab Bench. I have told you that you should begin to see that biology is no longer bench, it's no longer only on the bench, it's no longer on wet. You don't need to wear lab coats. What you need to do is how can you make sure you can use your computer, and that's why we call it in silico. In silico means inside computer. So bioinformatics is basically done in silico, that is with the use of a computer. And from what we have said so far, if you are talking of computer, you can actually make use of even your phone, because your phone is also computer. If you watch that video again, you will see that the first assignment, the first task, and which we are to do today, and to record in your practical tool book, that is workbook, is that you need to do what is called local align, that is blast. You will use what is called NCBI. You go to where we call NCBI, just go to Google. When you get to Google, inside that box just type NCBI. When you type NCBI, you will see it. Probably we will see it today. So a beginner's practical guide to decoding the software of life. Just like I told you, the journey ahead of us is number one, we need to understand the molecular structure, which is the masterpiece, which has to do with DNA. Then, after that, we'll now pick this DNA, we'll now collect some segments, we'll fragment it. you cut it into small fragments. But that small fragment is going to be set of nucleotide bases. It could be ACC, GGC, it could be in form of alphabet. And that's why we say we now want to know, when you fragment it, you now want to know each of these, which of this one is similar to organism in the whole database of gene bank, where we call the gene bank. So what we are going to do, and that's what we call blast, is that after we must have fragmented this, cut it into small fragment in form of nucleotide bases. It could be of just several bases. Now we want to compare that one to the whole of a gene bank, where there the information about nucleotides, where information about gene are being kept. So that's why they said you will align the code, you align them, you put them together. When you put them together, you now find the area of similarities and find the area of dissimilarity. Wherever you see the area of similarity, you record it as +1. Wherever you found the area of dissimilarity, differences, you record it as -1. So, and wherever there is a gap, we call it as a -2. We are going to see all this. Now, you now bring them together. What do we mean by alignment? Alignment is that you bring them together. And there are basically two types of alignment. We have what is called pairwise alignment. Pairwise alignment, that means you are bringing two nucleotide sets, you are bringing them together. Only two. That is, you can be comparing your set of nucleotide with another organism. That is pairwise. But what we now call multiple alignment is that you now bring one, which is your set of nucleotide. The one you have is called the query nucleotide. So you now want to see anywhere in the whole world which organism has something that is related to that. That's why I said you search the database, and that database is called the NCBI. We are going to see it, and that is exactly what you should do as part of your mini project. So, and that is what you are supposed to do. So, querying the global DNA. So, where you have the whole DNA of different organisms throughout the world. So, you have your set of nucleotide here. You now bring it here to see if there is any area of similarities or dissimilarities. So, in that video, you will see that the video was talking about e-values. So, e-value is telling you that if the value you get at the end of the day is very close to 1, that means your query nucleotide with every other one in the database, they have match, they have similarity, they are homologous, they are very similar, and that means the e-value should be tending towards 0. So what we are saying in essence is that the higher the e-value from 0.1, the further, that is, the more there is differences between them. So that is to tell you that if you have 3 organisms, when you have 3 organisms that you compare your query sequence with, if one is 0.001, another one is 0.002, another one is 0.003, you will see, because the first one, which is 0.001, is very close to 0, that means it is more close, it is closer, in fact it is the closest to your query. The three nucleotide that you are using to compare them, they are called the subjects. The one you have at hand is called the query sequence, and those one you are comparing them with are called the subject. So immediately when you now compare them together, so that will now help you to be able to plot the evolutionary tree, which is a phylogenetic tree that is connecting the data to the real world. And we will see all this as we progress in this uh, course. You are not just learning biology, you are learning computational skills. Once you are able to do this, it is no longer just ordinary biology, because it is biology that is done outside the laboratory. It is biology that is being done on your phone. It is the biology that is being done on computer. You are now thinking of an organism in form of codes, in form of letters, in form of nucleotide bases. So that is the essence of what we are going to do today. Okay, now we have talked about what is called the molecular trinity. That is the DNA. This is the DNA. That is the double-stranded ladder. So which consists of the bases. A will always pair with T wherever you see it. G will always bond with C. In fact, this journey actually started in 1953. The journey of bioinformatics started in 1953 when Watson and Crick were able to predict correctly the double helix structure of DNA. The basis, this basis A to T, G to C, is called the Chargaff rule. The Chargaff rule. So now I mentioned something about the Central Dogma theory the other time. The Central Dogma theory is the flow of information from the double-stranded DNA to single-stranded RNA, then to protein. Now you can see the RNA now, which is single-stranded, and this one is deoxyribose sugar, that is removal of oxygen from a ribose. A ribose sugar is a 5-carbon sugar. That's why they call it ribose. So deoxy, removal of oxygen. Ribose, that means it still contains oxygen in carbon 2. So, and apart from that, I told you the other time that instead, you can see here A to T, but instead of T here, A has been replaced by U. So, and this one, there are 3 basic type, very common type of RNA. These are the mRNA, which is the messenger RNA. The second one is a transfer RNA, and the third one is a ribosomal RNA. Okay, you must have learned this in your biochemistry, molecular biology, cell biology, and maybe biochemistry. So then this is protein. That is amino acid, set of amino acid. When you start arranging them, I told you the other time that there are basically 20 types, 20 amino acids, essential and non-essential. And these essential, when they are arranged on a straight line, when they have only one strand arranged in one straight line, that means that protein is existing at the primary structure. And whenever they are now folding, the folding could now be secondary. The more folded a protein is, the more functional that protein is. The fold determines the destiny of that protein. If a protein is incorrectly folded, that means that protein is going to have problem at the end of the day. And that is the reason why the structure of protein is called the destiny of that protein. We will see it as we go on, and you will see it from that same video that the difference between somebody who has a sickle cell, a sickle shaped red blood cell, and the one that has the normal ellipsoidal shape red blood cell is just in the folding, is just one nucleotide, just a replacement of just one nucleotide base, and that is exactly what has made the difference between somebody who is with hemoglobin A and somebody with hemoglobin with a sickle, that is a sickle shaped. So the cell. So we are going to see all this as we go on. So, but you should know that protein is called the biological machine. It's called the biological machine, and the reason for this we will see it as we go on. So, just like I told you, the central dogma theory and the sickle cell glitch, you can see a single mutation, that is just a single alteration in the letter in the DNA code alter the entire DNA, entire 3D structure of that protein. So the problem you can see there is difference between this red blood cell and this one. So you can read more about it as we go on. So then our DIY, that is do it yourself, uh, this, what you will do is that I want you to write this thing down. ATG. ATG is going to transcribe, that is on RNA. ATG is on DNA. So when it is transcribed, it's going to be AUG. And the interpretation or the meaning of AUG is going to be the amino acid that is called methionine. So you just process biological data manually. Then when you say CGA, the CGA is still going to be CGA in RNA, but it is arginine, that is the amino acid. So I want you to continue to sequence all this so that to write all sequences and be able to transcribe them from inputs, that is the DNA. Then what does it mean by in RNA, and the interpretation in protein. So don't forget that nucleotide, you don't forget that these are called triplet codon. Triplet codon. So you will see more of my video which were dedicated to protein synthesis, the genetic code, and so on. Okay, now I mentioned something, that there are basically two types of alignment. I've told you what alignment is. Alignment means you are bringing two things together. You want to straighten them, you want to compare them. Okay, compare two sequence by 1 to 1, 1 to 1. This is sequence 1, this is sequence 2. That is pairwise alignment. I've told you. So when you look at this sequence, you see that sequence 1 contains A, sequence 2 also A, like that. So that is what is called pairwise alignment. But when we talk about multiple alignment, that means you have more than two. And you can see when you now bring them together, you will see that all these four sequences contain C. So this C means that they are from the same ancestor. The same ancestor. That is why they have similar here. Not only in C, even this one also indicates that. GGGG also indicates it. This one also indicates it. So this one is telling us that they have similar ancestor. But wherever, when you compare sequence 1, sequence 2, where you compare these 5, you will see that instead of A to be here, you now have T. So this is a point of differences. So this point of differences. Now this is what I was trying to tell you. The question is, how does the computer interpret? Don't forget that computer does not understand alphabets. what computer understand is just code in form of zeros and ones. That is what the computer understand. So I told you the other time, wherever you have, when you bring two sequences together, sequence 1, sequence 2. So if they have the same, you record them as +1. T +1. Well, that's why this one is +1. This one is also +1. But here, you see, G and C, this is -1. We call it error of mismatch. That is -1. Computer will record it as -1. So, and this one is also +1 because it is this. But instead of A to be here, nothing is here. So this is a gap, and a gap is recorded as -2. So this is how, this is exactly what we call algorithm. An algorithm is nothing but the flow of one computer event to another. So that is algorithm. Scoring the alignment. How do you score the alignment? That is, how does the computer thinks? Okay. Now we are already going into what is called the data, the blast. Now this is your sequence. I told you that is called the query. Now you now bring your query sequence. You now do what is called BLAST, means basic local alignment sequence tool. It's like the Google. It's like the search engine that contains everything. So, and it is known as NCBI platform. That is the gateway. You now put your sequence here. From here, you check it from where it is called the NCBI. So the NCBI has contained lot of information about DNA. So when you are now comparing them, that means you are doing what is called BLAST. BLAST allow you to cross-reference your singular biological code against the entirety of known sequence life. So who will see it? Then I have also mentioned something to you. Also, this was also mentioned in that video too. The decoding the e-values. I told you that the more your final value is tending towards zero, you can see that 0.01 is higher than 0.001. So this is a strong match. This is strong match, highly significant, biologically related. So anyone that is tending towards 0.1 or 0, that means it cannot be an accident that they are similar. So you report them as 100% similar. So if they are tending towards 0, but whenever they are tending to be greater than 1, that is a weak match, and that is likely to be a random chance. It might not even occur. It might just be an hypothetical situation. So this is your confidence score. The closer the number is to 0, the stronger the biological relationship. And we as biologists, what are we actually trying to say? What are we interested in? Don't forget that when COVID-19 came, they were able to trace the family of COVID-19. That is, it was from SARS-CoV. Okay, so that will actually tell you. So the closer we are towards 0, the stronger the match, the more significant, the more biologically related they are. Now, this is what you will now do. On your phone, just go to your phone, go to your browser, go and type BLAST. Go and type in Google, go and type NCBI, or you type this. You enter this website in your mobile browser. So when you type it, something like this will come up. That is step 2. You will see nucleotide blasts. You tap the nucleoside blast module, then you will now enter this sequence, exactly this sequence, AT, GC, GA, TT, TG, AA. When you do that, you now run what is called blast. You hit the blast and wait for the query to run. Wait for it to run, and then you'll be able to record it. So just like I said, interpreting the digital products. So you will now see how closely they are in terms of e-value. I told you that if the e-value is closer to 0, that is telling you that 100% identity. They are 100% identity. And this can be used. You can see now that this is one of the reasons why there is a what you call bioinformatics is very, very important. You don't sometimes what you just need to do is that just get sequence, a raw sequence from your organism and go and correct, and go and compare it with what is. So now when you get it, let's say this the okay, let's just in an hypothetical example, let's say there's an outbreak of diseases in which nobody, let's say it is even a new disease, so which nobody has ever known anything about it. So what you will do is just collect samples from your patients. When you collect the sample, you do sequencing. Sequencing means that you synchronize, you break down the whole organism into their alphabet, and that's what is called sequencing. So that you now have them in form of ACTGGGG like that. I told you that that of TB is around 4.4 million or 5 point something million. That of E. coli is around, I think it's a little bit in thousand. That is in thousands. Okay, so you just pick any part of it. You just pick. You can just decide to pick 20. You can pick 25. You can pick more than that. So when you now pick that, you will now go and run what is called blasts. Similarity. You want to know diagnostically. You want to know which one, even if you do not know the name of the organism yet, but you want to know which organism it is similar to. And similarity means that they are going to come from the same family. People that are from the same family, they have more identity than people that are not from the same family. The more closer related they are, the more their similarity. So what we are saying in essence is that this e-value is going to tell you a lot about it. You use this one as a diagnostic tool. So you can use this to tell them as a bioinformatician that you don't even need to get into the laboratory to go and run trial and error. Apart from that, you will expose yourself to the risk of getting infected. You spend a lot of money on that. So what you will just do is to get this one, just sequence it. And don't forget that I told you in the first lecture about the successes recorded so far by bioinformatics, in the sense that in those days, to sequence the whole of human genome project costs more than $3 billion. But nowadays, if you want to just get the whole genome sequence of an organism, even with $200, you can get it done. And the more the numbers of nucleotides, the more. So E. coli, in fact, with maybe less than 100,000 naira, even in Nigeria, you'll be able to get the whole sequence of the E. coli. So that is to tell you that so many times, and any diagnosis that is based on sequences, based on bioinformatics, based on DNA, based on molecular diagnosis, it has no mistake. It has no mistake because it is going to be more specific. it is going to be, in terms of specificity, it is going to be more accurate than just trial and error, and at the end of the day we just even see that we are not able to. So the query sequence belongs to a bacteria, and the near zero value confirm an incredibly strong non-random biological similarities. Okay, now why it matters, the evolutionary record. Don't forget that I told you immediately after you must have gotten your e-value. The next thing is how can you make biological meaning out of it. So I told you the more closely related they are, the more they are very close to their ancestors. So, and all the roots, those things that make them to be those ones that make them, so those ones that make them to be those them that make them to be very similar means that they are very close to the ancestor. They are coming from the same ancestors. Okay, so just like I said, that we must be able to establish how closely related organisms are and how unclosely, that is unrelated, they are. So you can see now the ATGC is what has brought all of them together. That is why they have the same root here. And when you look at this one, this is going to be a mismatch, and the score is going to be what? -1. This one, the score is going to be +1. This is going to be +1. This is going to be -1 as well. Okay, so at that point in time, you will now see that they now have branched. That is why they are branched here, because of this unrelatedness in there. So similar sequences means the shared biological ancestry. Differences, that is mutation, means evolutionary changes over time. So bioinformatics allows scientists. That's why one of the reasons why it matters to be able to run what is called BLAST. That is, how related are they, how unrelated are they. Bioinformatics allows scientists to track mutation, trace diseases origin, and also map the relationship between all organisms. So this is what you are going to do, reports in your lab work, in your workbook. What are the aim? You will see in the workbook, aim, what are we analyzing? That is the aim. What are we analyzing? Aim, to analyze, to see how similar, to run blasts. That is what we mean by aim. The materials, what are those materials that are needed? Hardware, those ones that you can see, those ones that you can touch, and those ones that you need to do by the use of internet. You don't need to install anything on your phone by now if you want to run BLAST. So that is going to be your step 2. Step 3, the procedure you are going to use step by step. Step by step. I have mentioned that procedure for you the first time. Go to Google. When you go to Google, or you type that website in your browser, type it there. Then visit NCBI. When you visit NCBI, you paste the sequence there. When you paste the sequence, click on BLAST, then you record your this. So you should be able to record the step by step. Then the next thing is results. Organism. The result, the organism did you get? Which organism is it very close to? You list all those organisms that your sequences are very close to. Then, what are their e-values? What are their e-values? then the identity, their identity. So are they identical? How unrelated are they? How related are they? Then the interpretation. How will you be able to interpret? How will you be able to interpret by the use of a e-value? Then what are the conclusion that you need to make? That is the summary statement. What can you conclude on? What could be? What lesson can we learn from that as biologists, as bioinformaticians? What can we learn from there? So then the reflection, there will be a point in that your workbook that will be asking you, what is the reflection? What have you learned in this video? What have you learned so far in this practical section? What have you learned? What was so difficult that you could not learn, that you did not understand? List them. Then documenting the friction in your process. Documenting the friction in your process is where true computational thinking develops. So what we are saying in essence is, by the time you are able to follow out all these steps by step, you are able to run blasts on your own, run it successfully. That is telling you that you are no longer a biologist of yesterday. You are no longer a biologist that depends solely on wet lab. You are no longer a biology, but you are now a computational biologist that will work, that will be able to synchronize, that can reduce the whole organism into only set of data that you can use in your computer, in your laptop, on your phone, to be able to get all this. Permit me at this point in time to give you briefly about what I have been able to do and what the project that I'm still on. So I am actually working on multi-drug resistant mycobacterium tuberculosis. I am trying to get some immunogenic markers, that is, some of the sequences that they are using as their own weapon to fight against human, to be able to fight against all antibiotics, that is making them to be resistant against common antibiotics. So be able to get all those weapons against them. So let me just get that weapon in form of their alphabets. And it is in that alphabet that I will now make use to be able to design a vaccine against them. Is their own weapon. I want to use their own weapon to be able to fight against them. And not only be able to fight against them, what I'm going to do and what I'm willing to do is just to make sure that I trigger, I use their own weapon to be able to trigger an immunological response. That is, I trigger the immune cell of the body. I prepare the immune cell of the body to be able to fight against them at any point in time, whenever they see them. And that's what is called fascination. And I am using that. I've already started the wet lab. I use whole genome sequencing. I've gotten my sequence. My students know very well because we are also working together. So we have sequenced that. We got them in form of alphabet and this one and that one, and we have been working in order to be able to use it to design a vaccine. Because the vaccine that they are using against TB is TBCG, and that is more than 100 years old. And apart from that, it is no longer even effective. It is not effective in adults. It can only be used to control, just around 80% effective in children, but not even effective, just around 20%. To tackle adult pulmonary TB, not to even talk about, is not even effective against drug resistance. So that is what we are going to do. Now, just like I said, that bioinformatics practical reports. So now the course title you will see, this is exactly what I'm actually expecting you to do. The aim to identify an unknown DNA sequence using NCBI BLAST. That is going to be your aim. That is what it is expected. And the materials you are going to use include your phone, smartphone. Then you need an internet browser. Then you need a notebook. You need a notebook. This time around, the practical notebook I said you should do. That is a you should enter all this. I gave it to you. Yeah, the one I made the video for you. So you also make use of that. Then the procedure. What procedure are you going to use? Open NCBI. So you are going to select nucleotide blasts. Then you inputted this and you run the analysis. So the top match results, top match is equal to E. coli from this one. So you can also check this because I'm going to give you another sequence that you use for your own report. But cross-check. So whether you are able to get this. So top match is E. coli. The E value is 0.001. Then it has 100% identity. That is, the query sequence is exactly, it has the same similarity, it has the same sequences. So the interpretation, the sequence belongs to a bacterial genome. That is established. The near zero E value indicates a highly significant match. Then the reflection from this lecture is that manually we are translating the sequence first made the computational speed of BLAST much easier to appreciate. So if you are able to do this successfully, if you are able to do it and you are able to understand this, and that is exactly what I want you to do, be able to, so you'll be able to understand this, and that is the beginning of your journey as a bioinformatician, using only your phone, your smartphone, to do it. And having done that, you will see the beauty of molecular science, adding them together with data science. That is the future discovery, and that is the reigning currency, and that is nothing but to tell you that the future only holds, the future is only sure for those that are able and that can be able to run biology, to learn biology, to practice biology, to implement biology on not only in the laboratory any longer, but on the smartphone. You run it, you make your diagnosis on smartphone, make your presumption, your prescription, you make your prescription, make it based on that. That is why at a point in time we have just talked about bioinformatician or bioinformatics being used in drug design, being used in vaccine discovery or vaccine design, being used in agriculture, because it is now the biology. It is in silico biology. So you are not just observing life. It is no longer you are actively querying its source code. We have a lot of data that is available. So you should be able to run a live blast search who can find the homologous sequence with absolute lowest E value. And if you are able to do that, I am very sure, very, very sure, I am much more convinced that the future of data science, the future of digital biology is on our fingertips. It is tending towards personalized medicine, whereby the drug that is going to be produced is only going to be meant for you and for you only. That is personalization. We want to have a vaccine, some set of vaccine, pharmaceutical products that you will be tending, including diagnosis. that is going to be based on the sequences, uh, that is going to be precise, it's going to be personalized. So having said that, I think I want to thank you so much for being with us this evening. Don't forget what I've said, that I am expecting you to submit your workbook. That is going to be exactly 24 hours after that. You know where to submit. You're going to send it to the email I've just given you. Don't forget, this is EduInteractAI. You can still see us and see some of our digital products at www.eduinteractai.org.